I think that research in pediatric surgery involves all pediatric surgeons, open or minimally invasive. What I'm going to try to transmit to you is that we are not that good at that, and it is very necessary to change our minds and our approaches in order to give to research the importance it has in our specialty as in any other specialty of surgery. I'm going to address the two aspects of research. Clinical research first, probably it's more important for all of us, and basic laboratory research also, to show you that many things should be approached in the laboratory, as I will show you a little later. But first, clinical research. Our actions, our operations, our choices of treatment are mainly based on clinical research, which in fact is the way of analyzing on the basis of our experience the results of what we do. And there are many ways of doing that, but in fact for the majority of our operations or for the majority of our acts, we have been always acting on the basis of observations of our past. And this is a biased way of looking at the things. If I am only doing a Swenson technique, well, I publish on that and I say that this is very good. But people doing Duhamel technique publish at the same time their results and can also be very good. And there is no way of really objectively defining which is best for the patient. And that's what we have been doing, and we are still doing all the time. You have seen that in this Congress. Many people have presented different approaches that are based on personal observations or the history of we have done in one or other service. And this is a not very objective way of doing that. Fortunately, well, this has been the same for all other medical specialties, but in the last 25 or 30 years, there is a progressive trend that becomes invasive to change the approach to this clinical research. And we are going more into more objective things. Let's say in oncology, this has been very clear, establishing protocols that are practiced by several institutions or including several countries, have been able to put together a large number of patients and to judge of the benefits of this or this other treatment. Randomized controlled trials are the way of defining in a relatively short period of time which is better for this particular disease or for this particular operation. Unfortunately, as you will see, this is not that easy in our specialty and in other specialties, but particularly in ours. I've taken some time to show you the articles that were published in the main 3 pediatric surgical journals on research. And you see here that, in fact, this is the Journal of Pediatric Surgery from in this decade, from since 2002-2012, and you see that only about 20% or a little less of the articles published in the journal were research articles. But if you look at the clinical research articles, that randomized controlled trials, meta-analysis, you see that the proportion is really very small. And if you look at the European Journal of Pediatric Surgery, you can see that more or less we have the same thing. Out of 100 and something articles relating to research, only a few proportion are scientifically approached clinical research articles. And also is the same for Pediatric Surgery International. You see that a very small proportion of these articles were scientifically sound approaches to clinical research. And this is something that should concern us because there must be reasons for that. Many years ago, 1998, Valeria Varandini, who is currently in Milan, as you know, was working with Agostino Piero and Lucy Spitz in Great Ormond Street, and they decided to assess how many of our operations are based on evidence-based arguments. And you see that according to what they did, only 26% of our operations are based on randomized controlled trials. And basically, the vast majority of what we do, operations, were based on what we call eminence-based decisions. That means that they are based on the analysis of follow-up studies or retrospective case studies, as all of you have done or are still doing all the time. So the thing was very weak at that time, and recently this has been reanalyzed by a group in Oxford that published recently, a few months ago, in PLOS ONE, this article that may seem difficult. You see, what evidence underlies clinical practice in pediatric surgery? Systematic review assessing the choice of the study design. They designed for a study 2 years, separated by 15 years, first 1998, and they analyzed in the literature 50,000 articles by pediatric surgeons or on pediatric surgery, and they could demonstrate that basically half of them, or almost half of them, were retrospective series, as I have mentioned before. But you see that another 39% were case reports, 3, 4, 15 case reports, but that's it. And then there are small numbers of K-control studies, a very small number of randomized control studies, 2%, and some prospective cohorts that are better as an evidence, and some long-term follow-up, and a very small number of systematic reviews. And you may think that in the ensuing 15 years things have changed very much, but in fact you see that in their analysis in 2013, you see here, the number of pediatric articles was much more important, 3 times more or 2.5 times more, but you see that again 50% of these articles were retrospective series. The proportion of case reports didn't change, and the only thing that changed substantially is a certain number, a growing number of systematic review, which is a quite good evidence that it's a very small number of articles, and that didn't change very much. So, if you want my opinion, we are not very good at clinical investigation. And that explains why a group doing only colonic interposition for esophageal replacement thinks that this is very good, but the group that is doing gastric pull-up believes that this is the best way of doing that, and the only a few people that are doing jejunal interposition thinks that this is very good, because no one is comparing with objective methods the different techniques, and it is certainly difficult to do that. Why is that in our specialty? In our specialty, this is worse than in other specialties. That is mainly because we treat a large spectrum of conditions. It's true that we are the real general surgery. We have everything, the thorax, the abdomen, urology, the tumors, and whatever. And obviously, it's very difficult to act on a so large spectrum of conditions. The larger spectrum is accompanied necessarily by the small size of the series. So it's very difficult. Well, among you, I'm sure that not many people have more than 10 esophageal ascopticians a year in their hospital. And obviously they are treated by 5 or 6 doctors. That means that the series are always small, and that ends up with multiple learning curves with a small number of cases. And even worse, we have to assess our results many years later. Let's say continence, or it's not a question of survival or not survival, it's a question of quality of life, of many things that are only known many years later. And also we are, and this is very good, continuous innovators. When I was training pediatric surgery some years ago, I learned Swenson technique. I came to Madrid where Swenson technique was also practiced, but I learned with Suave himself, who came here several times to operate, that his technique was very good, and I moved into that to do that in younger children, and then all the other innovations, including Luis de la Torre innovation, and we have had to learn everything in a relatively short period of time, so that makes multiple learning curves, small series, not many cases, and on top of that, ethical limitations, because our patients are not autonomous, and they are difficult to include into randomized controlled trials like adult patients. So we have ethical limitations that are shared by pediatrics, of course, by branches of the science that are devoted to children. So clinical research is very imperfect in our specialty, and we have to do something about that. And then basic laboratory research. You may think that this is very far apart of your interests, or the interests of some of you. But in fact, I don't believe that. I would like to try to convince you that it is necessary to develop this thing also within the specialty, for the following reasons. The first is that we don't understand most of the diseases we are treating. Let's say necrotizing enterocolitis or the lung problems of congenital diaphragmatic hernia. Nobody understands that very well, and if we don't understand, we are not going to treat that properly. And on top of that, nobody really are interested in these conditions except ourselves. So go to basic scientists or even pediatricians, they are not interested in what we treat. We should be interested in what we treat, and we should do something to solve our question marks that are so many in our specialty. This is my institution, some of you know it. This is Hospital La Paz, this is the Children's Hospital, this is the Maternity Hospital, and that's the General and the Orthopedic Hospital here. This part and all that have been transformed now in a research facility. This is medical school that I am just across the road. And this, in this center we have, along the last 25 years, developed a quite strong research facility that is shared by all the services of the hospital, not only pediatric surgery. Currently, the Institute of Research of La Paz, they have one very similar in this hospital, has a payroll of about 180 people, and we have a quite large budget devoted to research because we believe that this is very important. We have done that keeping the old ways of research for the pediatric surgeon that are used for training as well, operating rooms, microsurgical places, but also we have developed all the necessary laboratories for molecular and cell research, and this has been done putting together all the different projects of the different departments of the hospital, and I think it has been quite successful along the years. And as regards my group, I have been fortunate to have the collaboration along the last 35 years of people from many countries. The majority of them, I'm not going to point them, all of them, which ones are pediatric surgeons, but the majority of them are pediatric surgeons that decided to devote some of their time, a couple of years in general, to develop some research, eventually a PhD dissertation in our place. And I have been very fortunate to have extremely skillful people that work with us in the different projects in pediatric surgical research. But I insist they are mainly pediatric surgeons, and I think this is something that is very important and very rewarding for me. Our idea has been that the research should ideally go in this direction: disease to basic research, molecules and genes. And when you look at the scientific literature, you see that the other way around that doesn't work, or usually doesn't work. Many of our malformations are present in the basic studies of basic scientists, but they are not interested in that. And they don't develop the whole thing. And I think that we have to go from the disease to the molecules and not the other way around. And I am try to illustrate that with one of the paradigms of pediatric surgery, that as our research on esophageal atresia in the last 35 years. We have also worked, as some of you know, very much in the mechanism of gastroesophageal reflux, in the way of understanding why that happens and so on. We have devoted a lot of work to congenital diaphragmatic hernia and also to small bowel transplantation. But in fact, I'm going to summarize only our experience, our studies on the esophageal atresia that you know very well, and of which we have a lot of experience. You see, these are the data from 1998, We have now about 600 and something cases, and we have studied that with a lot of interest in the origin of this malformation. And for that, we have used a model that we created ourselves. It's not true, but it was in the basic science, in the basic toxicology science, because when adriamycin was studied as a cancer agent, they described that rats treated with that had pups with esophageal atresia and other malformations, but nobody gave attention to that until our friend Juan Diez Pardo, who was the one with Dr. Chi, the Chinese fellow that was working with us at that moment, retested this thing and described this magnificent model that allows to reproduce the malformation. You see, this is a neonatal rat dissected from behind. You see here the upper pouch of the esophageal atresia, the trachea, which is much longer than in human individuals, and you see here the tracheoesophageal fistula going into the stomach. The model is identical to the human disease, and the proportion of associated malformations, the same we see in the clinical setting, are demonstrated here. 50% of our cases had esophageal atresia, but many other malformations were present, as in the Vactor association. These are sections of normal rat fetus at term, and you see here, this is the pharynx and the larynx, and you see at this level, you see already the esophagus and the trachea separated, you see the thyroid here, and if you look at the sections of one of these animals with esophageal atresia, you see the same larynx, and you see how the esophagus disappears progressively at the different levels until disappearing totally. Here you have only the spine, and then you have the trachea, and that's it, until you go down, and here you have the fistula and both lungs on both sides. Exactly the same thing we see in the human individual. And also with Dr. Chi, we described that there were islands of respiratory epithelium in the lining of this esophagus. The same thing we see in a human individual. So we reproduce exactly the malformation. But then we went into some other things. We decided progressively to understand why this happened, how atresia is produced. And then we went into basic studies, and you see here summarized that esophageal atresia is something is wrong in the separation of the trachea and the esophagus, and you see here the foregut from which both of them arise. You see here the notochord, and you see here the posterior part of the foregut in yellow, and you see here the anterior part of the foregut in blue. This will represent the respiratory part, and this the digestive part. And here you have the mesenchyme that surrounds this part that will become the lung with all these canals here. And in here you have a number of genes and transcription factors that are in charge of directing all these operations, particularly Sonnich-Hedgehog that acts here, and also in the interaction with the mesenchyme, but also NKX2, that is TTF1, or FGF10 that is here. I'm only going to address this one. I'm not going to tell you all the research we did, but we were interested in this particular thing, and from the beginning we were interested in the action of the notochord in this thing. And the main thing we found was this is the neural tube in an embryo treated with adriamycin and with esophageal atresia, and you see that the notochord is split in the sagittal plane. There is something that is abnormal in the notochord, and the same thing has been found in the mouse model that was developed after that, and we have many reasons to believe that the same thing happens in a human individual. Look at that. This is what we know all from the clinical setting. You see, these are all patients with esophageal atresia, and you see that many of them have more or less important anomalies of the spine, wedge vertebra, split vertebra, very distorted spine, as you see here, together with some malformations of the limbs that you know well. Well, here I show you a normal rat fetus at term to show you how we have imaged the spine, and you see in red is the bone, in blue is the cartilage, and you see the limbs in here. And you see that 100% of animals with esophageal atresia have either wedge or split bodies. The same thing we see in human individuals. And also some of the malformations of the limbs recall what we see in the clinical setting. The same thing for I'm not going to go into the detail on that. The heart malformations and the great vessel malformations. I'm just going to show you some of the great vessel malformations that are typical or typically found in cases of esophageal atresia. You see that mainly the coarctation of the aorta, right aortic arch, aberrant right subclavian artery, and so on. And I'm not going to show you the heart because it's more complicated, but look at that in these animals that, by the way, had often absence of thymus. You see aberrant right subclavian artery, you see this one passing behind the trachea, double aortic arches or right aortic arches, the same way we see in human individuals. So we're probably dealing with the same mechanism of anomaly. Then looking more directly into the respiratory tract, with Sandra Monteadónico from Chile, that was working at that time in our lab, we analyzed in 415 cases of esophageal atresia, we analyzed the respiratory tract malformations, and we found them in 6% that were in fact not often detected in the survivors, but were found in a sizable proportion, 13% of those who came to autopsy. So that proves that probably they are much more frequent than we think. And that makes sense because certainly some of them were very relevant, but others are less relevant. Right pulmonary hypoplasia, left single lobe lung, esophageal lung in very rare cases, of the right tracheal, big bronchus that you see in a number of cases with esophageal atresia. So it's not that strange, and we went into the lab to see why this happened and how this happened. This is a tracheobronchial and lung block from a 13-day-old normal fetus rat, and you see that put into culture, this thing branches and branches until 72 hours later when you see almost a lung that is formed in here. Well, when we treat these individuals with esophageal with adriamycin, we have this interesting shape. You see the fistula in between both bronchi that you see here, and you see how branching proceeds in the same way, but the end result is not as brilliant as in the normal individual. So less branching, and in those animals that were treated with adriamycin and didn't have esophageal atresia, you see that the bifurcation, the carina, is totally different, and also you see that the branching is scarce as in individuals with esophageal atresia. So we moved into looking at what happens with the number of terminal buds. That is a way of knowing how much branching we have had, and you see that along these days, 0 and 72 hours of culture, you see how in comparison with controls, both groups treated with adriamycin had at least 50% less branching than the other animals. So that means that probably we are into a problem with development of the lung. And you see here, look only at this, if we use the same criteria for lung hypoplasia that we are using for CDH, for congenital diaphragmatic hernia, the lung weight body weight ratio in percent, you see that animals with esophageal atresia have definitely lung hypoplasia. Of course, not the same lung hypoplasia as CDH animals, that will be probably here, but some sizable and statistically significant lung hypoplasia that is also illustrated by the radial alveolar count, that it's a way of measuring the branching of the airway. You see that both groups treated with adriamycin had less branching than the control animals. In other words, we have some degree of lung hypoplasia. And then we went, and I finished with that, into trying to understand why this happened. And then I recall you that this is the epithelium and this is the mesenchyme, and you see this is sonic hedgehog is expressed in the tips of the epithelium, while patch, the receptor here, is expressed in the mesenchyme. And you see here that sonic hedgehog acting on patch unchains smooth, that unchains the production of the GLI proteins, and these act on other things that are in charge of developing the lung. And so with Dr. Fragoso from Porto, who spent a long time with us for her very good PhD dissertation, demonstrated, I'm not going to show you all those things, that at the embryonic, pseudoglandular, canalicular, and saccular stages, successive stages of development of the lung, sonic hedgehog was changed in these fetuses treated with adriamycin and having esophageal atresia. You see here, this is significantly smaller than in control individuals, and you see that along the gestation, this reverses, and at the end, there is some probably compensation by overexpression of sonic hedgehog trying to regain the control of the development of the lung. This is probably relevant also for understanding how that happens in the human situation, and I think the only way of studying that is through laboratory effects. I conclude first on the clinical research. I want to point out that solid clinical research in pediatric surgery will only be possible if we put together our strengths, if we collaborate between or among several hospitals and probably among several countries in order to produce large numbers of patients to judge in a relatively short period of time of the benefits or setbacks of the treatments or the ways we are treating our patients. And obviously, prospective studies are the only way of advancing in that. And I urge you to collaborate with other people in order to put together our strengths and work synchronously with other people. And as regards basic laboratory research, should be encouraged. That doesn't mean that you all should go into that, but remember, no one will be interested in our conditions unless we take in charge this investigation. That doesn't mean that we cannot collaborate. We may motivate other researchers. We have to communicate our interest to basic scientists in order to motivate them. But if we don't do that, in 20 years' time, we will be still guessing why necrotizing enterocolitis happens, or what's the mechanism of the pill in gastroschisis and so on. We have to be involved in basic science, and those of you who have the material or the necessary arrangement, you should go into that, and you should try to motivate young people to spend some time doing basic research, because I think it's the only way of advancing in our specialty. And again, I want to thank you, Philippe. I know that this is probably a little different from what you expected from François Laborde, but I hope that I will have given you a view of what is basic research in clinical and basic research in our specialty. Thank you very much.
Thank you very much, Juan. Thank you for this great lecture. Thank you for the great kindness for us to be here today with this lecture. And I would like to remember you are the very founder of the first European society, the European Association for Pediatric Surgery, and it's a great honor for us. And may I ask a standing ovation for Professor Thomas. Thank you.